Hello, everyone. I hope you are all doing well and staying healthy. My name is Nicole, and I'm a junior biomedical engineering major. I'm also in the Accelerated Master's program. Today, I'm going to be talking about the research I've done since I became involved as a research assistant in Dr. McGinnis's lab. This semester, I conducted a survey of activation models from studies that use electromyography signals to drive muscle contraction dynamics under my graduate student peer mentor, Reid Gerchiek. First, I'm going to talk about what a neuromusculoskeletal model is and how my main focus has been those modeling activation and the roles that they play in muscle contraction dynamics. Then I'll talk about my RU, which involved my initial literature review and the origin of 3 activation models that were a result of that. Then I'm going to talk about how these findings led to the need of a formal literature review that I'm still currently working on. And lastly, I'll touch on my additional research experiences that I've had. Neuromusculoskeletal models are used to define the relative location of bones and muscle surrounding joints. In human biomechanics, the aim of these models is to be able to estimate or predict muscle forces and joint moments from neural signals or EMG measurements. In a research paper written by Thomas Buchanan, it can be described as a 4-step process. In the first step, muscle activation dynamics govern the transformation from the neural signal to a measure of muscle activation. In the second step, muscle contraction dynamics characterize how muscle activations are transformed into muscle forces. You may know that when you multiply a force by its moment arm, you can get the moment. Similarly, the third step requires a model of the musculoskeletal geometry to transform these muscle forces into joint moments. Finally, the equations of motion allow joint moments to be transformed into joint movements. This process is known as forward dynamics, where you compute motion from the known internal forces. The opposite of this would be known as inverse dynamics, in which internal forces are reconstructed from the motion. The focus of my research has been models in that first step, activation dynamics. Neuromusculoskeletal modeling is important for studying how the nervous system controls limb movements in both unimpaired people and those with neuromuscular disorders such as MS, Parkinson's, and Huntington's disease. One of the leading trailblazers in modeling musculoskeletal structures is Scott Delp, a professor at Stanford who developed the open source software known as OpenSim for biomechanical modeling, simulation, and analyzing human motion. The picture on the right is an example of a simulation someone made in this program of the lower extremities. Next, I'm going to give a brief overview of the role muscle activation plays in muscle contraction dynamics. In order to use EMG signals in muscle contraction dynamics, we must first transform them into a muscle activation. This is where models come in. Activation models consist of a neural excitation, or EMG input, and an output of muscle activation. The term muscle activation is actually associated with the calcium activation of the muscle contraction process. This activation then energizes the cross bridges of the motor units to generate muscle force through muscle contraction dynamics. In the top figure, you can see calcium plays a key role in muscle contraction. The muscle contraction cycle is triggered by calcium ions binding to the protein complex called troponin, pictured in green, which expose the active binding sites on the actin. This gives way for myosin to form cross bridges with the actin and contract the muscle by attaching and detaching to pull the sliding filaments past one another. So, now to put variables to it, in the bottom figure you can see the neural excitation going through activation dynamics with an output of muscle activation, which then powers muscle contraction dynamics to generate a muscle force. This figure is also from the Buchanan paper. I think it is helpful to see because it also shows the joint kinematics and moments that can be estimated or predicted. On the left is the input EMG signal, output activation, which then drives muscle contraction dynamics, by which then you can estimate muscle force, and given musculoskeletal geometry, you can estimate joint moments. The joint moment is the sum of all of the musculotendon forces multiplied by each of their respective moment arms. With activation, we can estimate or predict muscle force, and with muscle force, we can then estimate or predict joint moments, given the musculoskeletal geometry. So, musculoskeletal geometry determines the moment arms of these muscles. When all of the muscles of any extremity pull on the bone, it makes them torque. Just because you've figured out what the net moment is from inverse dynamics does not mean you know what the individual muscle forces are. There could be 10 or 15 different muscles pulling on any bone at once at any given time, and we don't know how much each one is pulling. This is the fundamental problem for researchers studying muscle activation and contraction dynamics. Like I mentioned before, the main focus of my research so far has been neuromusculoskeletal models describing muscle activation, specifically EMG-driven models. The fact that it is EMG-driven is important because EMG can be measured with wearable sensors. The use of electromyography signals is becoming more common in wearable sensor-based remote patient monitoring and gait analysis. EMG-driven muscle models may provide estimates of more clinically relevant biomarkers. This semester, I had the opportunity to complete an REU, which is the research experience for undergraduates. I conducted a survey of activation models from studies that use EMG to drive muscle contraction dynamics. The purpose of my research was to understand the scope of EMG-driven activation models that have been used for neuromusculoskeletal modeling and their origins to consider application for remote gait analysis. The initial phase of this project focused on studying research papers and articles that define and use EMG-driven muscle activation models, and then tabulating the information about how it was done. The results of this initial analysis was the origin of 3 known activation models. Like I mentioned before, as the use of EMG signals for remote patient monitoring is becoming more common, this information may be used to consider their applications for wearable sensor-based gait analysis. It is extremely important to validate and understand what currently exists in literature so we can make informed decisions when it comes to choosing the appropriate model to use. The preliminary phase of this project focused on reading and classifying the papers included in the review recently published by Ursula Trindler in 2018. Earlier, I talked about how the fundamental problem for researchers right now who are studying activation-contraction dynamics is the inability to determine the individual muscle forces from an estimated joint moment. This review identified a bunch of different techniques specifically applicable to clinical gait analysis, such as static optimization and inverse dynamics. It included several papers with EMG-driven and forward dynamic modeling approaches. This is where I started my search. After reading each of these papers, I classified whether they modeled activation or not, and if they didn't explicitly say what type of activation model they used, my process was to go to the methods section of each paper and look at the papers cited there. I went to read those and continued to work backwards. I found that most activation models originate from one of 3 papers: Zajac, 1989, He, 1991, and Milner-Brown, 1973. This is the first activation model I read about. Identified in Zajac's paper from 1989, Muscle and Tendon, Properties, Models, Scaling, and Applications to Biomechanics and Motor Control. It is a bilinear first-order model. You can see that activation, A, is on one side, and neural excitation, U, is on the other. C1 is the activation time constant, or how quickly the muscle activates, and C2 is the ratio of activation to deactivation time constants. A muscle tends to activate faster than it deactivates. In a fully excited muscle, the neural excitation is thought to be at its maximum, and activation dynamics is assumed to be at its fastest. In the references is an unpublished analysis, also written by Zajac, and that is how we came to the conclusion that this is what the model was based on. This paper is unique because it broke down concepts such as contraction dynamics that were still fairly new in research at the time this paper was published. When people teach these concepts in classes, there is really no textbook per se, so people reference publications and articles like this one. This was the first of its kind to summarize many different approaches and techniques, and it's easy to read. It was the first paper I really started with, and I was able to teach myself a lot from it. So, if you're looking for a good introduction to biomechanics, I would definitely recommend reading this one. Interestingly, Scott Delp, who was the creator of OpenSim, which I mentioned earlier, did his PhD under Zajac. This is the second activation model I read about. Identified in He's paper from 1991, feedback gains for correcting small perturbations to standing posture. It is a nonlinear first-order model. For a while, I originally thought that the origin of this model was in a paper that was published almost 20 years later, until I found this one in the references of that paper. They don't provide a citation for it. It seems to me that they manually constructed the model themselves to agree with 3 physiological features that they state in the paper. 1, that activation follows neural excitation asymptotically. 2, it has a larger rate constant for activation than deactivation, which keeps to the experimental evidence that muscle force rises faster than it decays. And 3, it is an analytical function suitable for feedback control analysis. This is the third activation model I read about. Identified in Milner-Brown's paper from 1973, the contractile properties of human motor units during voluntary isometric contractions. It is a linear second-order model. I think it may be considered to be the most justified model because it is supported by physiological evidence. Basically, the neural activation depends not just on the current level of neural activation, but also on its directly recent history, or the last 2 values of U. The discretized version of this model is very popular in current techniques. I now have a running list of papers that explicitly use and cite this model. Here is a comparison of the 3 activation models against a raw EMG signal for the soleus when you apply it in MATLAB. You can see that the green line is the raw EMG signal, the orange line is the Zajac model, the pink line is the Milner-Brown model, and the blue line is the He model. The He model most closely resembles the raw EMG. From my REU, I found 3 activation models and their origins. I am currently working on a more formal literature review to confirm these results. The purpose of creating a more systematic database search is to confirm that the studies that use EMG-driven muscle activation models only use or cite one of the 3 approaches that I described earlier, or if there is one that I haven't seen yet. I am answering these 3 questions. What models have been used and which ones are the most popular? Where did each model originate from? Meaning, who was the first person to use it, describe it, or develop it, and how did they do this? And how do I quantify which one is the best model? I ended up answering the first 2 questions with my REU. To evaluate which model is the best one, we would have to know which one yields the best results for estimating joint moments and movement. This is really up to the discretion of the researchers using it. This is all being done with an eye towards being able to model activation with wearable sensors. Much like the Trindler review, my paper will describe the approaches that have been used so others can make informed decisions when choosing a model. The paper will aim to summarize what models are used, in what contexts, and their origins. The databases I used were IEEE Explore and PubMed. When doing a structured search, it's important to use as little search terms as possible so you're not pigeonholing yourself. My search query included studies that use EMG data, activation, and gait analysis. My inclusion and exclusion criteria was that it must include experimental EMG data, whether they used surface EMG or intramuscular, so it wasn't simulated. I also only wanted studies that were only done on humans, so no animals. I also eliminated all conference papers because they are not as strictly reviewed, unless a model originated from a conference paper. Then I will definitely include that one. The most important part was that they had to either provide the activation model they used or provide a reference for one. If they didn't model activation dynamics at all, then the paper was rejected. The result of my initial search was 134 papers. I was able to exclude 55 papers just by reading the title and knowing that they were unrelated to what I was looking for or would not agree with at least one specification of my inclusion-exclusion criteria. Conference papers were also included in these 55. This left me with 79 articles to read and classify. When I'm reading the articles, I am looking to make sure they pass the inclusion-exclusion criteria. All of them are interested in neurological disorders, musculoskeletal diseases, and motor control. I only need to read the abstract and methods closely to determine what activation model they use, if any. So far, I've found that models have either directly stated or cited one of these three approaches that I found earlier, or cited another paper that cites one of the three. Or they do not model activation as a differential equation at all. In some cases, I've found that researchers have gotten the surface EMG envelope and assumed that was the activation. In another case, researchers got the surface EMG envelope and used it as an input to the activation model. I've been working through reading articles for about a month now, and most of the time the model is not clearly referenced. I often have to go back through a few references for the same paper, and then go through the references of those papers, and sometimes go back even further through the references of those papers. Outside sources are papers that fell under this category. In my search, I recently came across a new model. I found this paper by backtracking references for one of the articles in the IEEE search. It was constructed by Song and Horry in 2015 from their conference paper, Muscle Activity Estimation Utilizing State Observer and Neuromusculoskeletal System Model. It is a linear first-order model that is similar to the Milner-Brown model, except this is first order and theirs was second order. This was published only 5 years ago, so it is really new compared to the others. It is the most recent use of this model that I know of. It is possible that it appeared in an earlier paper that I may find by reading more papers down the road. I am interested to see if there are any recent studies published in the last 5 years that have used this model. From Song and Horry's methods, it seems to me that muscles are assumed to be stimulated by positive feedbacks from joint torques for their proposed neural input activation model. So, to sum it up, now we know 4 existing neuromusculoskeletal models of muscle activation requiring neural input from the nervous system. E91, a nonlinear first-order model, Zajac 89, a bilinear first-order model, Milner-Brown 73, a linear second-order model, and Song and Horry 2015, a linear first-order model. In conclusion, as the use of EMG signals in wearable sensors is becoming more prevalent in remote-based gait analysis, knowing the extent of activation models to exist is extremely important. I believe validating and understanding what already exists in literature is also exceptionally important. It has been very useful to me to be able to know how to backtrack the references in literature. This skill has helped me in all of my other research projects that I've done this semester. There's really no textbook per se that sums up everything that's ever been discovered or every technique that's been used in human biomechanics. People often teach and learn from reading literature. I've learned a lot. This is still a relatively new science. From what I've accomplished so far, I know that a discretized version of the Milner-Brown model appears to be very popular in current techniques. This model was published almost 50 years ago, and I believe that with current advancements in technology, there may be a reason to revisit muscle activation models in future research. In addition to my own research, I assist in data collection for healthy subjects by working with participants and troubleshooting the equipment during testing sessions in the MSense group. I also help by post-processing the motion capture data in Vicon so it can further be analyzed. I enjoy being an integral part of the process and seeing it through to the end.